Welcome to the virtual Stickleback Evolution Lab by Howard Hughes Medical Institute. Before we proceed, let us first define evolution. Evolution depends on the process of natural selection, and it describes a change in the characteristics of a species over several generations. Basically, it is based on the concept that all species are related to each other and gradually change, due to some factors that might occur over time. Stickleback was discovered to become a great model for studying evolution. These are very small, silvery fish at least 2 inches in length. Also, it is mainly a spiny fish consisting of dorsal, pelvic, and anal spines. So what's the deal? Studying evolutionary processes is essential as it explains various similarities among vastly diverse forms of life, any changes that might occur within populations, and even the development of new life forms. In this virtual laboratory, you will be introduced to the science and techniques used to analyze the forms and structures of organisms, in particular, the pelvic morphology of the thrust-beam stickleback fish. Furthermore, it will teach you skills of data collection and analysis to study evolutionary processes. Let's start. First, go to the virtual Stickleback Evolution Lab by Howard Hughes Medical Institute by clicking the link provided in the laboratory manual. On the screen, you can see that it is divided into 2 parts. On the left side of the screen is the virtual lab where you do all the experiments, and on the right side are all the information to guide you through the simulation. Click the virtual lab to enter. A note will then appear that welcomes you to the virtual lab. Below the note is the button to start the first tutorial, so click that button. In the first tutorial, you will be practicing how to score the preserved and stained stickleback fish. To begin, click the tray of fish as mentioned. Move the cursor of your mouse over the fish to magnify and inspect the presence of pelvic girdles and spines. There are 3 categories of pelvic structures: complete, reduced, and absent. Complete means that the pelvis has a full pelvic girdle and 2 pelvic spines. Reduced means a range of structures from a simplified girdle with no pelvic girdle spines to an oval nub for a girdle. Absent means that there are no pelvic girdles and spines present. You can change the view from lateral to ventral view to the structures present in the fish. After scoring the fish correctly, you will then proceed to experiment 1. However, you will have the option to skip the tutorial. Experiment 1 is divided into 3 parts. The first part is the fish staining. To start, click on the blue gloves. Then, click the jar that contains the fish from Bear Paw Lake, and click on the jar to see an unstained fish. Click the strainer to drain the jar of fish by pouring the alcohol into the empty jar. After that, click the flask of alizarin red staining solution to submerge the fish in the jar. Let it sit for 3 to 6 hours. The dye stains the bones red to make the pelvic structures much easier to see. Click the timer to continue. After staining, pour the staining solution into the waste container by clicking the jar of fish. Click the destaining solution to cover the fish in the jar. Afterwards, empty the jar of fish with a destaining solution by clicking it. Destain the fish twice, but the second time, submerge it for 1 hour, then pour the destaining solution again in the waste container. Next is to rinse the stained fish with water by clicking the jar and pour the water out into the waste container. Repeat again, but on the second time let the water sit in the jar of fish overnight or approximately 12 hours. Click on the timer to continue and pour the water afterward. Check the stained fish by clicking the jar. Add back the 50% isopropyl alcohol to the jar of fish by clicking the alcohol jar. This ends the staining process. Click the jar of fish to store for later use. Following the fish staining, let us now proceed to part 2 of experiment 1, which is the scoring of fish. Do this by clicking Go to Part 2, Scoring button. To begin, click the jar of stained fish from Bear Paw Lake. Pour out the alcohol by clicking the strainer. Notice a label was in the jar with the fish, it must stay in the fish at all times. Click the label to continue. Do the fish scoring just like in the tutorials section. Once you are ready to score the fish, click the pile of fish to select and view it under the microscope. When scoring a fish, click the pile of fish and view it under the microscope. Once done, click the microscope to place the scored fish in the other tray. After scoring 20 specimens, click the jar to put the fish back into the jar. Score another set of fish from Frog Lake by clicking the jar labeled with Frog Lake. After scoring both samples of fish from different lakes, complete the table in your notebook on the left side of the screen and click the Submit Total button. Let us now proceed to the last part of Experiment 1, which is graphing your data. Steps on how to graph the data are listed in the notebook. In this experiment, a bar graph will be created to visualize the results. Simply click the underline click here and choose 2 for the automated version of your graph. After generating your graph, go back to the virtual lab and click Verify your data. Take the quiz. After the quiz, proceed to an analysis by clicking Experiment 1 Analysis. A Chisquare test is used to analyze the data. Fill the table using the data you gathered plus the data given from Marvola Lake, then click Submit. Fill in all the other tables given to generate your Chisquare test result by following the steps in your notebook. Before proceeding to the next experiment, you must know first how to score a fossilized stickleback fish by performing the tutorial. To start, click the tray of fossil specimens. The guides in determining whether the fossilized fish have complete, reduced, or absent pelvic girdles can be seen in your notebook. There are also 3 parts pertaining to Experiment 2. Part 1 is the preparation of the fossils. To start, click the laboratory bench on the virtual lab, then put on your goggles and mask to protect you from dust and small pieces of rock. Next, click the tray of rocks with fossils to view it under the microscope, but before viewing, click the probe to slowly scrape away at the rock to reveal the fossil. Once the fossil is revealed, click on the fossil to proceed in Part 2. Part 2 is the scoring of fossilized fish. The same method is applied in Experiment 1. Click the tray of fossils to begin scoring. The first is to score Layer 2 fossils, the next is the Layer 5 fossils. Once done, fill up the table in your notebook, then click the Submit Total button. Afterwards is the graphing of the data. This time, a line graph is created to visualize the results. Simply click the underline click here and choose 2 for the automated version of your graph. After generating your graph, go back to the virtual lab and click Verify your data. Take the quiz. After the quiz, proceed to an analysis by clicking Experiment 2 Analysis. This time, you will calculate the rate that best describes how quickly this change occurred in the population. So, using your data, calculate the rate of change in the relative frequency of fish with a complete pelvis for 1,000 years by getting the relative frequency of the stickleback fish. Now let's move on to the last experiment of this activity, which is to analyze the reduced pelvis rate in more detail by scoring sticklebacks from 2 freshwater populations according to whether the pelvis is larger on the left or right side. This experiment is also divided into 3 parts. To begin part 1, click the gloves. Do the staining process just like in Experiment 1. Since you already know the staining process, you can proceed right away to part 2. Part 2 is about scoring the pelvic asymmetry of the fish. Click the jar of stained fish from Bear Paw Lake first. Strain to remove alcohol, then pour the fish onto the empty tray. Click the pile of fish from the tray to view it under the microscope. There are 2 outcomes in determining the asymmetry of the pelvis, one left bias, which means that the pelvic vestige is larger on the left, and the other is a right bias, which means that the pelvic vestige is larger on the right. In scoring the fish, select the pelvic asymmetry score by clicking the buttons below the specimen. After scoring 20 specimens, click the jar to put the fish back into the jar. Score another set of fish from Coyote Lake by clicking the jar labeled with Coyote Lake. After scoring both samples of fish from different lakes, complete the table in your notebook on the left side of the screen and click the Submit Total button. Afterwards is the graphing of the data. A bar graph is created to visualize the results. Simply click the underline click here and choose 2 for the automated version of your graph. After generating your graph, go back to the virtual lab and click verify your data. Take the quiz. After the quiz, proceed to an analysis by clicking Experiment 3 Analysis. A Chisquare test is used to analyze the data. Fill the table using the data you gathered then click Submit. Fill in all the other tables given to generate your Chisquare test result by following the steps in your notebook. That's all for the virtual Stickleback Evolution Laboratory. Thank you. Now it's your turn.